All right. Hello, everyone. Thank you for the opportunity to speak here. My name is Nicolas Escobar. I'm a PhD candidate at Indiana University. Um, and today I'm going to tell you a little bit about basically brain imaging. And so, because this is sort of a general purpose conference, let me begin by telling you a little bit about fMRI imaging. And so, the way this works is that we put people through scanners, right? And the scanners measure something called a Bolt signal, and they capture a signal for each little piece of your brain surface. And we basically, when we do that, we basically have two questions. You can ask when a subject is performing some kind of task, what parts of their region of their brain activate, and the other question that you can ask is even when they're not doing something, when they're so-called resting state, how do different parts of their brain interact with each other? And that's the question I'm mostly interested in. And the way people do that nowadays is through something called connectomics. So the idea behind a connectome is that you put someone through the scanner gives you a Bolt signal for each little part of their brain. You can think about that as a very high dimensional time series, and you want to summarize that data. And the way you do that is you just compute the covariance matrix. Because of the properties of covariance, what you get out of that is something called a symmetric positive definite matrix, or SPD matrix. And sort of the typical one is you can visualize on the right. And people try to do basically two kinds of things with that. They can either try to infer a brain network out of the connectome and then just study the network. Or the other kind of thing that people do is that if once they have the connectome, they try to get some biomarkers out of it to predict some conditions or to analyze how people age or stuff like that. And so the fundamental problem that I'm concerned with is the fact that the set of SPD matrices is a very special set because in particular it's not a linear subspace, right? If you add two SPD matrices, you may not get another SPD matrix, right? And to be more precise, that set is a cone inside Euclidean space. And so the practical implication of that is that if you want to compare two connectomes, you have to do it carefully, right? So originally, what people used to do was they would get the connectome and they would just vectorize it, right? Just get the entries of the connectome, put in a long vector, and compare vectors that you obtain from different patients. And that would be equivalent to sort of measuring the distance, the blue line distance, right, in Euclidean space. But you can see that sort of ignores the geometry of the set, right? You should be looking at distances across sort of the cone. And so what we want to develop is geometry-aware statistical methods for this kind of sample. So the one way to do that is through ideas from an area of math called Riemannian geometry, right? So the Riemannian geometry, what it boils down for us is you have your set, which we also call a manifold, of the SPD matrices. Then at each point in the set, you have a plane that sort of just touches the manifold. It's called the tangent space. And you can go back and forth between the manifold and tangent space through maps called the exponential map and the logarithmic map. And you can do something else, which is, as I said before, it's a bad idea to just vectorize the connectomes, but it's a less bad idea to vectorize the tangent representations. And so you get these three different representations of connectomes, either in the manifold or in tangent space, or as a vectorization of the tangent space. And this is all by now sort of an old idea. This has been thought about and developed for roughly 40 years now. My particular interest is in multi-sample connectomes. So what's happening is that actually connectomics studies have relatively small sample sizes because it's just so expensive to put people through scanners. So we're trying to get larger studies, but if you're going to do that, you're going to have to do it multi-site, right? You can just not collect 10,000 people in the same site. But if you do that, you're going to have to deal with the problem that the scanners themselves have a little bit of a difference, right? They can be the same model and the same make, but they're never quite the same. And so you're going to have sort of systematic differences between the data that you collect in different sites that is not biological, right? And so in that scenario, there are two things that you might want to do. The first thing is to test whether those differences are statistically significant, right? And the way to do that is basically through ANOVA, but you have to be careful about how you do ANOVA. And the other thing that you may want to do is to harmonize, which means to get rid of those systematic differences, right? So the key abstraction for this is this sort of diagram that I have here, where you're going to have individual samples that you collect at each site, and then you're going to have the pooled data, which I'm going to call a super sample, right? And sort of this tracks with ANOVA, right? Because when you perform ANOVA, you have sort of between things, and then you have within things, and then you have between things, right? And so there are some calculations that you perform per sample, and then there are some computations that you perform on the whole thing. And so this gets me to introduce the first package, which is called riemmtan. And it's the first R implementation of Riemannian statistical methods for multi-sample analysis, right? And so you have, as I said, you basically want to do two things, either ANOVA or harmonization. When you try to do ANOVA, there are actually a couple of ways to do that. So you cannot just vectorize the connectomes and do ANOVA on that. That's a bad idea. But there are things that you can do, and the sort of the established method is something called Fréchet ANOVA. That's a very popular method. And then you can do that. And then me and my advisor were introducing a new way to do ANOVA for this kind of data that can be more powerful than Fréchet ANOVA in certain regimes, but it's also computationally more expensive. In any case, we implemented that. And then for harmonization, there are actually a couple of ways that you can do that. The first one is called COMBAT. This is a very popular framework in genomics. And it can be adapted for this kind of data. And then you also have something called rigid harmonization. So this is a snippet of how riemmtan works. You're going to have those three lines there. You're going to create sort of objects for each site, right? And then you pull them together in this super sample object. And once you have that big object, you can just call the necessary function that you're interested in, right? You can do Riemannian ANOVA, and you can do it with either Wilks lambda or Pillai's trace or all of those typical things. But you can also do Fréchet ANOVA. And as I said, there are advantages and disadvantages. Okay. And then for harmonization, again, there's COMBAT. COMBAT is an empirical-based framework. And the nice thing about COMBAT is that not only does it allow you to get rid of the systematic differences, but you can also preserve the differences that you do want to keep, right? It's possible, for instance, that at one of your sites, you collected data from older people than in other sites. You would like to keep the variation due to age, but not to site. And so that's something that COMBAT can do. And then there's rigid harmonization, which is a little bit cheaper computationally. And we have implementations for both of those. So that was riemmtan. But then we said, okay, riemmtan it's all about multisample, right? But if we want to do multisample, at some point we have to do computations at each sample for one sample things, right? And there are packages that do that. So there are a couple of them. One's called Riemann and another one is called Fréchet. And they do everything that you want to do for single sample things. But we thought we could improve on that because there are a couple of limitations that we considered to be quite serious. And the big one for me is that the Riemannian metrics are hard-coded into the package. So there are actually multiple ways. If you have a Riemannian manifold, there are multiple ways to measure distances, in particular for SPD matrices. And the thing about it is that people keep coming up with new metrics all the time, right? And so if these packages, of course, both of these packages are very flexible in that they have implemented a bunch of metrics, but there are new ones and new ones. So we thought that Riemannian metrics should be sort of modularized out of the package. And the other big problem that they have is that they're all serial, right? And we thought that we should do parallel things because connectomes are very high dimensional, and so any computation takes a long time. And so if we can do things in parallel, that would be better. So that's why we decided to also mess with the one sample things, and that's what riemmtan is all about. And here's a snippet of what riemmtan can do. So riemmtan is the one that is going to give you the single sample object, and it's an R6 class. That's why you have the syntax there. It's going to have some methods. For instance, it can compute the Fréchet mean. It can center the sample and compute the vectorized representations. And you're also going to have your active bindings, right, for R6 classes. But again, the thing that I consider key is the fact that that the metrics are sort of separate sort of modules. So you can define your own metric, and in order to define a metric, you don't just have to tell me how to measure distances between 2 points. A Riemannian metric is a little bit more than that. You have to tell me how to compute Riemannian logarithms and Riemannian exponentials. But once you and also how to perform both vectorization and the opposite, the inverse. But once you do that, you can just put it in your sample object. So to summarize, what we have is 2 packages. We have riemmtan that does the single sample computations, and it is very generic in that sense. It has sort of the core algorithms, the thing that anyone who does this kind of thing wants to do, and it's sort of a foundation layer that we want people to use, that we want people to build on top of. And then separately, there's riemmtan, which is a package that does the multi-sample side of things, specifically harmonization and ANOVA. And just a word about the underlying sort of structure. These are as you could see from the snippets, these are all based on R6 classes. We do try to do parallelism using the parallel package, and then there's also for the heavy matrix sort of computations, we do have a C++ part of it. Here is a benchmarking slide. So this is for something called computing the Fréchet means of a sample. So if you're in a Riemannian manifold, or in particular, if you're in the set of SPD matrices, you cannot compute means just by averaging. Or you could, but you're not going to get the thing that you want. Because what you want from a mean is not that you can add things and divide by n. What you really want from a mean is that it minimizes square distances, right? And this notion of this notion of minimizing square distances can be generalized for something that doesn't have a linear structure. But as I said, Fréchet means are defined as something that minimizes distances, which means it's an optimization procedure. And for SPD matrices, it can take a little bit of time, right? And it is also critical, right? If you're doing statistics, you want to compute means all the time, right? So it's sort of this very core piece of software where you use it all the time, and also it's very expensive. It can be very expensive. In any case, we have here on connectome size and sample size, and you can see that riemmtan just on wall clock time significantly outperforms Riemann. So that was basically it. Riemmtan is already on CRAN. Riemstats is just on GitHub. We have vignettes, and let me just acknowledge the Lilly endowment, which helps with the high performance computer at Indiana University. Thank you.